CMU. The talk will be at 3:30 on the 10th floor, and it will be followed by a reception as we always do. And the title of Alex's talk will be Epistemic Diversity, Ethics, and the Optimal Timing of Clinical Trials. All information is on the website, so if you want to know what's coming on in the coming weeks, not just next week, please have a look at the calendars on the website. Today, it's my great pleasure to introduce Lotem Elber-Dorozko. Lotem, many of you know Lotem because she's been at the center for now a bit more than a year. She got her PhD at Hebrew University of Jerusalem in computational neuroscience. So she has a foot in neuroscience and a foot in philosophy. She was a postdoc, and she's still a postdoc, which is last year, and she is now a visiting fellow at the center. We're extremely lucky to have Lotem with us. She has published in excellent journals in philosophy and in neuroscience, including cognitive science recently on representation and cognition, about whether neuroscientists can ask the wrong questions. She also has a recent paper in a synthesis about the role of mechanisms in neuroscience. So really a lot of work that's really of interest to many of us. And today, Lotem will be talking about how in a field rife with controversy, some neuroscientific models become generally accepted as good models. Lotem, floor is yours.
Thank you very much. I'm very excited to be here, and thank you everyone for coming. This project, I think I have a good question that I'm going to present to you. I'm not so sure about my answer. So I think it's a perfect opportunity to present it and to have a discussion about the possible answers to consider for this. It's going to be a very sciencey project because I'm trying to use examples that I know from neuroscience to ask kind of my own philosophical questions. Oh, and also, if you have questions during the talk, please feel free. It's always more fun for me to have kind of a discussion while I'm giving the presentation. So just to give a short roadmap, this is last time I gave a talk here in January, and I tried to convince you that there are a lot of controversies in disagreements in neuroscience. I'm going to start from there and present some examples. And then I will present the standard methods that are not able to come above the controversies. They still yield controversial results. And then I will say, okay, but there are a few models in neuroscience that are in consensus. But generally, everyone agrees these are good models. For example, and this is new and exciting work, the ring model for representation of head orientation in the fly. I'm going to ask why is it convincing? I'm going to argue that it's the anatomical relationship to the function. And I'm going to make kind of an argument by elimination. Why are we convinced by anatomical properties? Do they indeed provide evidential support? Or is it just a kind of our own biases? And I will argue that they do provide evidential support. Present some objections to this one. If there's time left, I will give another example of a model that's in the consensus, which is about interoral time difference calculation. Interoral is the difference between the 2 years in the brainstem of birds. And finally, shortly some conclusions. Okay. So let's begin. Neuroscience has many computational, sometimes competing models, just very, very general. We can have different models for visual processing. We can present causal models, predictive processing models, convolutional neural networks. All of these are kind of competing models. Also for decision making, we can think about value-based models with diffusion, Bayesian inference. These sometimes compete, sometimes complement each other. We also see a lot of controversy in neuroscience, a lot of disagreements. So I thought I will have to work hard to convince you that there's a lot of disagreement in neuroscience, but if you're on Twitter, you may have seen the controversy. this is work that's became pretty famous in the media, adversarial collaboration to critically evaluate theories of consciousness. So this got a lot of hype, and then some people signed a letter disagreeing with the results. They integrated information theory of consciousness as pseudoscience. I don't have a lot to say about this. I try to stay away from theories of consciousness. But I would just say pseudoscience is maybe a bit too strong. But anyway, you can see that there's a lot of disagreement. To give you examples that are not about consciousness, about things that are a bit more basic in cognition, consider the method of using goal-driven deep learning models to understand sensory cortex. This has become very popular recently with the improvement in machine learning. We build a machine learning model that's able to classify images from pixels. So this is like kind of the blueprints of the model. They found a model that performs well. And then what we do is we correlate it to activity in different areas of the visual cortex according to what we know about processing in the visual cortex. And this has had much success. Has gotten also a lot of hype. Also controversial, deep problems with neural network models in human vision. This was published in PBS. The argument is basically that they're not explaining the right things about human vision. Published in Nature, neurons in the orbitofrontal cortex encode economic value. This is related to decision making, to evaluation. Again, much success. This paper has become very famous. And we also see disagreement, the case against economic values in the orbitofrontal cortex, so anywhere else in the brain. Okay, one final example. I know this is a lot. The Bayesian brain, the brain is Bayesian, the role of uncertainty in neural coding and computation. Bayesian tell just so stories in psychology and neuroscience. So, okay. What is going on here? I think is a bit bothersome. Like, I don't know. I'm trying to ask, this is what I asked last time. Is it the case for other sciences? But what we see is that people, they get results, they do the science, they record neuronal activity, they look at behavior, and they publish the results, and people still disagree with the results. They say, no, like, I don't believe you, right? I don't buy into the results. What are the standard methods that kind of still yield this controversy? Okay, so just to give one famous example, which is kind of easy to see. So I can, some people undergoing surgery, we can record from single neurons in their brain. So this is what they did in this paper. And they just showed this participant different images. And I don't even need to do the statistics here. What we can see very clearly, or maybe not because it's a bit pixelated, but images of Jennifer Aniston, right? So right here. And you see every dot here is like an action potential. It's like a neuron firing, exhibiting activity. This neuron really responds strongly to Jennifer Aniston, and it doesn't respond to anything else. Is it a Jennifer Aniston neuron? Do we have single cells that encode this type of information? Controversial. Even though the statistics are amazing. Okay, here I showed you before single cells, but usually we don't want to explain single cells. We want to explain activity in entire brain areas or networks. So what we will do is we will use activity in a whole layer in the artificial model, do linear regression to explain the variance in an entire brain area. And here we have like IT, IT is a brain area, inferior temporal cortex, explain the variance. These are previous models. They're not very good. This is the model that Yamins and Nikawa used. It does much better. Is this how we process visual information? Still controversial. Also, people, like when I talk about the issue of identifying representation, computation, there's also some discussion about correlation and causal relation. Correlations don't show causal relations. Today, we can get causal relations in the brain, and we can do this with methods that are very sophisticated. This is like a complicated picture. All I want to show here is that we can inject here the Tonegawa lab, they injected mice with viral vectors. So when they did field conditioning in a specific context, they were able to capture the cells, only the cells that were active during the field conditioning. And then a day later, they put the mouse in a different context and they only activate the cells that were active during that field conditioning. And even though the mouse is in a different context, see that when we activate it using light, when we put the light on, the mice exhibit freezing behavior, which is related to fear. So we can get these very complicated causal influence on neuronal activity and behavior. Did we capture the engram for fear? Did we capture memory? Controversial. Okay, so far about the controversy. Now some models that are widely accepted among scientists. So for example, the ring model is a fly, which I will talk about more. Jeffrey's model for computation of internal time difference for sound localization, which I will talk about if I will have time. Neural integration in the vestibulo-ocular reflex, encoding of sound in the cochlea. This is pretty much what I was able to find. Unlike other well-established findings in neuroscience, these models answer how capacities are achieved, not just what the brain is doing. So you can say we know very well where is the auditory cortex, visual cortex, motor cortex. It's not, I'm talking here not just about saying like where things are happening, but how they're happening. And that we still don't have answers for in many cases. So why do these models receive a status of being found? Perhaps not always the same answer, but I think it's still worthwhile to try and ask this question. So just as a qualification, before I begin, of course, you all also know history of science. This is also a question of historical narrative, right? So we know that acceptance of models can depend also on sociological factors, subjective factors, fads, fashions, having the right timing, having the right zip code. For this talk, I put these questions aside and I try to find other relevant factors. And I think maybe you'll disagree, but I think that for the models I present, their appeal, you will understand their appeal, and I think it transcends social factors. The ring model in the fly. So first, let's talk a little bit about head direction representation. Why do we need a representation of head direction? So let's say I'm a fly, I want to go in one direction and I accidentally tilted a little bit to the right. I want to be able to correct course. Right? So I want to be able to know where I'm heading relative to some location. So it's assumed that for different animals, they have a representation of head direction. The representation, of course, will be on a kind of ring, just like head direction is. And we expect it to have the following features. First, we need a representation for every angle of head direction. Second, we expect to see a single bump in the representation, right? I can't have my neurons representing that I'm both going north and going south. We expect this bump, the representation of a specific direction, to stay in place when the animal stays in place. Otherwise, it won't be able to consistently represent head direction. It should follow integration of angular velocity. So if the mouse moves clockwise or counterclockwise, it should be able to re-encode the representation, shifts according to visual cues. And we do indeed find in many different animals neurons that are responsive to head direction. And we find these in various brain areas. Now, let's talk a little bit about the ring attractor model. It's a very abstract model from dynamics. So the idea is this: how do I represent orientation or angle? I have here different states. Each state represents a different orientation, and states that are nearby, that have similar angles, excite each other, and states that are far away inhibit each other. And this model, we can write out the equations, and people have done so. With certain parameters, only states with a single bump are stable. And we see that we can also get inputs to this model according to angular velocity or visual inputs. We can re-encode the head orientation, head direction, according to the new inputs that we receive with this model. Now, I think it's important to point out about this model that it's about the represented features. So here, each dot is a different state representing a specific orientation. It can be, it looks like each dot is maybe a neuron or like a node or something, but there's no commitment in this model for that. Right? The model can be, I can have kind of different states across the populations. All that matters are the dynamics of the model, that I have stable states across a ring which corresponds to environmental property of head orientation, and that we have excitation for nearby angles and inhibition for far away angles. So this is not like a mechanism. It's just a modeling dynamics. Okay. So now a little bit about mechanism. So this is the central complex in the drosophila fly. And we can see it has different anatomical structures. Specifically, what we find is that we see a ring, right? This is called the ellipsoid body. It's ring EP, real short. And let's look at what happens when we record activity in the ellipsoid body while the mouse is walking on a tethered ball. Just to make the movie a bit clearer, we see here kind of a visual presentation that moves together with the fly that's walking on the ball in different directions. And we see activity, the nice kind of neon lights are calcium imaging. We see it basically they represent activity of neurons in that area while the mouse is walking. And we see that there is a very strong correlation between activity in the ring and the direction. These are like, they are compass neurons. So they represent, if the mouse walks this way, they will, the location of the representation will move the other way around. But basically what we can see is that there is a very strong correlation between the activity of the neurons and the head direction of the fly. To give some statistical data, so this is kind of the original paper that identified these properties. They calculated different properties of the activities that they found in different flies. We see that for everyone there's around one bump. There's one bump. It has a similar width. So like the width at half of the maximum activity. Strong correlation, PVA population vector average. Strong correlation between the direction that we get from the encoding of the neurons and the actual head direction of the fly. And you can get different, like the ring can be mapped in different ways to the external properties. If anyone has any questions about the science so far. Okay. Just to give, this is another example, and here you can see what we expect the head orientation to be from the neurons. This is the neuronal estimate versus what we see from the pattern position, from the movement of the fly. We see that they're very strongly correlated. So here again, you can see it across time, very close population vector activity to where with fly heading. And this is the activation. We see a single bump representing a single orientation. Okay, we also have causal information from interventions. So for example, if this is indeed an area that represents orientation, we expect that if I excite the neurons at a different like angular representation, then I will get a different, I won't have two bumps, right? I expect to see mutual inhibition. What I will have is only a single bump, and this is indeed what we see when the scientist excited neurons in a different angle to the one that was represented originally. And the bump remains in place. And also I can change the visual properties discretely. According to the properties of the model, we expect the representation of orientation to change continuously. I think this is actually a pretty interesting property because it's in cognitive science and models, we talk about inductive biases. So you can think about this model as implementing a kind of inductive bias. Orientation doesn't change discretely, it changes continuously. And this is what we see, the bar position changed discretely, but the representation of orientation by the model changed continuously, in line with what we expect from orientation representation in the brain.
Just tell me how the patterns in the EP are tracked real time while the fly is moving. So how did they measure? How did the people measure the EP region? So
the fly is tethered, and there's a camera attached to its head, and it's moving on the ball. But while the fly is moving, what's moving is the ball. And then we have virtual reality that moves according to the movement of the fly, but the head of the fly, in fact, remains in place for the recording.
So the head is not.
So it doesn't move relative to the room, but just like for the fly, its entire environment moves. People also search for connectivity features, and we see connectivity features that are in line with what we expect. From a ring model, excitatory neurons tend to relate to themselves and to nearby. So this is like 6 is like one slice and 5 is another slice. They relate to nearby neurons, which is what we expect from excitation. And we also see inhibition that is across different areas, general inhibition. So maybe you disagree. Philosophers sometimes disagree with scientists. But I'm convinced. I think how can it be anything else, right? With all the data that we have. But I just told you like everything is controversial in neuroscience. So why is this uniquely convincing? And we can suggest different things, different ideas. First, you can say this is an easy question in the sense that it's clear what the function has to be to represent head direction. You just told us you need to have a single bump. It needs to change continuously and with velocity. So it has to be that. It has to be this ring model, right? It can't be anything else. You can also say, look, you just showed me an integration of evidence that is very strong. both correlational and causal. You can say we have connectivity. This is pretty new. In neuroscience, we have such specific connectivity, and it's informative, and we don't see it in other cases, in most other cases. But what I will argue is that it's about the anatomy. And it's about the anatomy because anatomy allows inference of function. So I'll go quickly about why I think the first three are wrong. So we can say it has to be a ring model, so that's why we're so easily convinced. And indeed, I think some functions heavily constrain the computations that can underlie them, right? So we see this easily. If you want to compute position from velocity, it's just the physics of the world that we have to perform integration. So of course, what we will compute is integration. And if we want to detect edges, we should compute, maybe not necessarily, but it's useful from what we know about the physics of the world to compute second derivatives and look at zero crossing. This is from Mao. But let's look about the case of head direction cells. So we can compare what we see in the fly to what we see in mammals, right? And we also find head direction cells in mammals. We find them in a variety of anatomical areas, and also mixed with other types of neurons. Grid cells, places, cells that represent location. So when we look at what happens in the rat, we do find these head direction cells, angular velocity cells. We see that activity in these cells is in line with what we expect. It's persistent, it does not habituate. We see that there is tethering between the two head direction cells with different angles. But we don't find the head direction area, and we don't find obvious organization of the cells like in the ring, in the fly. We don't have excitatory connections, and we don't say that we found the ring attractor in rat brains, even though we have head direction cells. So we have here something that's kind of performing the same function as neurons in the fly, but we're still not convinced that we have the right model. Correlation and causal evidence is strong. I think it's strong, it's very convincing, but correlational and causal evidence we have everywhere. And we have models that are just as strong, and they're still controversial. Like there's not anything here that's qualitatively different. And it's not, I mean, it's strong, but it's not. This is very close to one, it's very impressive, but it's not overwhelmingly strong. Okay, connectivity is informative. Yeah, if we just had the connectivity, I don't think anyone would be convinced. Like it's very general. It's in line with, but it certainly doesn't support the ring attractor model. Okay. So I think it's the anatomy. In the fly, we see that we can map different roles in the ring attractor to neuronal types in different anatomical locations. So here we see, this is the type of neuron, and the ellipse of E denotes like the presynaptic, the input, and PG is the output location. And these are mapped into head direction cells. We have rotation like velocity cells, visual cells, vestibular cells. So we can map each type of neuron and the function that it has to a different anatomical area. And we also see an orientotopic organization, right? It's not that just these neurons represent head orientation like salt and pepper, like intermixed, right? There's a very strong correlation between the location of the neuron and the angle that it represents. So I think we see this bump moving on the ring, and we say, I mean, it has to be head direction representation. But this is maybe a bit surprising because, okay, anatomy is not connectivity, right? It doesn't tell us anything about the causal role or the causal effects that these neurons have on each other. Why does it matter? Specifically, why does it matter that in the fly, head direction cells are neatly organized, while in the rat they are dispersed and in various brain areas? And we can suggest 3 possibilities. This is where I get more into the philosophical part of the talk. Some people will say you're convinced, but you're misleadingly convinced. Right? You got a nice picture and an explanation that's like nicely wrapped in a bow. We're convinced because of our biases. It's easier to interpret and to understand. There's no real evidential support in the anatomy. It's just an easier explanation to understand. Some people may say that anatomy is pragmatically useful, right? So I have all the cells in the same area, so it allows observation and manipulation that are not possible otherwise. But it's just the pragmatics. There's nothing about the anatomy itself that helps evidentially. But I think that anatomy actually provides evidential support for function. It helps distinguish spurious from robust purposeful activities. So let's talk about these different options. Okay, so some people will say being swayed by anatomy reveals subjective bias. But anatomy does not actually tell us something new. It is a mistake. I think that anatomy allows us to infer functional units. So if we have kind of a thought experiment, let's say that we find a single neuron that responds to head direction. We say, okay, it may be indeed a neuron that represents head direction. But it may correlate with head direction because it represents some related property, right? Such as location. It may correlate with head direction because of some connectivity patterns that occurred without relation to its function. So it may be that it accidentally receives input from another neuron that represents head direction. And let's think about what happens when we think about the population of head direction cells in the rats. We say, okay, maybe this is a functional unit, but we say it may be part of a more general network dedicated to a more general function. Maybe there is no head direction representation as a functional unit in the rat. Maybe there's just representation of location, and head direction cells are part of that computation. And it may be divided into 2 or more functional units with different purposes, right? I can say maybe there's in the visual area, we have the what and where areas. Maybe we have head direction representation for movement and head direction representation for localizing objects. But when we look at the central complex of the flag, when we look at the ellipsoid body, right, it's much more likely that this brain area's response reflects a single function. Okay, so you can say maybe anatomy is relevant, but it's pragmatically relevant. It allows us to do experiments. Because we know where to find the neurons. I agree, it allows us to do experiments, but it's not just because it allows us to find the neurons, it's because we treat this anatomical structure as a functional unit. We say that all and only the neurons in the ellipsoid body take part in this head representation together, right? So I can find a dispersed population. I can find the engram, like in the causal experiment I described, and I can intervene on it and I can record it, but I just won't be convinced that I caught the entire relevant functional unit, right? Maybe I missed some and maybe I intermingled different functional units. So what this comes down to, I think, is that neuroscientists are in the business of distinguishing spurious and incidental results from results that indicate functional organization of the brain. So what do I mean by this? We can find correlation with neuronal activity that does not have causal power. So we say, okay, it's not actually the function of this neuron in the brain. We can say, I found this correlation, but it's the result of the system's encoding of some other property or function, right? So people can say, okay, we have confounds here, right? I see the encoding of Jennifer Aniston. It's very impressive. Maybe it's just the case that Jennifer Aniston pictures tend to have more luminance. I can explain variance in IT with the model that I presented, but maybe I can also explain it with many alternative models. And like I said, maybe the head orientation neurons in the rats are the results of computation that involves grid in place cells, right? So it's not, they happen to correlate with head orientation, but it's not their function to represent head orientation. So another option, which is kind of with a question mark, what happens if I find a neuron, it correlates with an environmental property. It has a relevant causal role, but it is the result of random changes in connectivity. So I happen to find in the fly a neuron outside the ellipsoid body. It also functions exactly like a head orientation neuron. I would say, I mean, it's very interesting. And maybe philosophers will be right to say that this neuron is computing and has that specific function, but I think it's outside the scope of current neuroscientific practice, which is in the business of understanding brain organization and function in general. So which result will indicate a purposeful organization? I want a robust result across participants with few degrees of freedom for interpretations. I want a case where I say, what else can it be? And I think this is the case I'm less sure about. I think when we look for these properties, when we look for robust results with few degrees of freedom, what we are looking for actually for purposeful organization are activities in organization that are likely the result of long evolutionary processes, right? We want the things that didn't just accidentally happen. We want the things that have a long history and reflect a general functional organization. So anatomy allows us to infer function. Because the ellipsoid body is a single anatomical structure, we infer that it is a functional unit, right? An area for head direction representation. And because of the mapping between angle and position in the ring, the kind of orientopic mapping, we infer a unique function of every neuron. So how do we infer function from anatomy? There are two parts. First, anatomy is a very strong constraints, right? It restricts the degrees of freedom scientists have in assigning functions to neurons. So if I have an anatomical structure that has a function, scientists cannot pick and choose, right? All neurons in an ellipsoid body are head orientation neurons. And the exact head orientation represented has to fit the position of the neuron on the ring. It cannot just be assigned to fit neuronal activity alone. And I think this is also makes the result much more convincing. And less convincing research projects do not have these constraints. But anatomical constraints are not arbitrary, right? It also indicates the history of the system. Well-organized brain areas are much less likely to be incidental or new, and are more likely to reflect a consistent organization of the brain. And that's why they are more convincing. So I'll just finish up with a few objections. People can say anatomy is just a proxy for connectivity, and that's why it's convincing. Okay, so anatomy and connectivity, I think, are closely related, but it's interesting to consider what happens when they come apart and what will convince us. So connectivity changes during our lifetime. It differs between individuals, and it is more fine-grained than anatomy. So let's say that I have a neuron that, due to connectivity properties in the ellipsoid body, doesn't fire in accordance with the model. I think we will still take the function of this neuron to be a head direction representation, even though the connectivity is not in line with that. And we will take this function to be head direction representation because we think the anatomical structure reflects a function that came about through long evolutionary processes. And let's imagine a group of neurons in an individual. There's some idiosyncrasy. They have come to correlate with head direction, have causal effect. as I said, I don't think we aim as scientists to explain their role in head representation. We will say it's interesting, it's incidental. So what I will say about this is that anatomy is kind of a blueprint for connectivity. It's a rough guideline for what connectivity should be. And connectivity can build on that. Possible objection, function should not be defined according to history. I think this is the first time I'm citing philosophers in this talk. It's a long time. So I think philosophers make good arguments that function, we can define function not according to its history. Cummings talk about systems functions, or describe a function according to the function of the general system that it's part of. Piccinini describes forward-looking functions that have goals but are not dependent on the history of the organism. But I think, and maybe you disagree with the intuition, but I think this approach resists the intuition that the anatomy of the ring model is informative. And also, I think that this way to view function may be a good description of ontology of functions. But when we want to identify functions, what we need is to look at historical processes. Finally, this is an objection that I received a lot when I kind of talked about this idea. That is, okay, it's great that you found the ring model in the fly. It's completely irrelevant to what happens in the cortex, especially the cortex of people. Right? In the cortex, we see there's population encodings, there are manifolds, there's context-dependent computation. Computation is non-localized. So there's no way to translate the organization and the ellipsoid body of the fly to cortical activity. What I say to this is, I think if researchers present one result for the entire population, then they say, look how this manifold is different from that manifold for a population of hundreds of neurons, then that is indeed much less convincing than the results that we see in the ring model, in the fly. But there are possible remedies. I think that we can learn from this model. First, we can examine variance between neurons and between participants. So we know that scientists like to look at averages. That variance matters because if I see small variance, it demonstrates consistency, which is what we're looking for, right? We want to see consistent functions and consistent organizations. And this is maybe a bit out there, but I would say that maybe instead of focusing on neuronal activity, which is what the vast majority of scientists do, we should focus more on organization principles. And this will allow us to put more constraints on our results and to have more convincing results. Rather than just finding a significant correlation, even a strong correlation, and saying, look at what we found. I wonder if I should I go into the second example or?
About 15 minutes, or good 10 minutes up to you.
Okay. I was a little bit tired.
I'll go first.
Okay. Another example for a good model that we found in Neuroscience. So as an introduction, we know that for many animals, it's very important to localize where sound is coming from. For example, owls capture their prey by localizing like the sound of the mice or the rats. And there are different ways to do this. Generally, what they do is that they rely on differences between the sound that arrives at the 2 ears. You can do this by having a different at levels, interneural level difference, like how strong the sound is. Now, if the frequency is very high relative to the size of my head, then I will hear it, it will be much weaker when it arrives to the further away ear than the closer ear. So that's one way to do it. But if the frequency is low, I can't rely on this level difference. I have to rely on time difference. So I have to compute the difference in time for the frequency at which it arrived each year. How do animals do it? So one kind of intuitive model that Jeffers came up with is this idea. We have delay lines coming from each ear, ipsilateral, contralateral ear, and then we have coincidence detectors. So what happens if I have a sound that's coming right in front of me? It will take the same time to arrive at both ears, and then I will have, if I have the delay lines that take the same time, which is here in the middle, the coincidence detector will fire most strongly. Coincidence detectors, they fire when I get inputs at the same time from both ears. Now, if I have a sound that's coming from the left, it will arrive more quickly to the left ear than to the right ear. And then the coincidence detector that takes a longer time with the delay lines from the left ear than from the right ear will fire, and so forth. So each coincidence detector, you can think about as a neuron, and it represents the delay between the two ears, which corresponds to the angle of the location of the sound. So this is the model that was suggested. And it's actually pretty astonishing that we see exactly this model. Not in all animals, we don't find it in mammals so far, but in birds, in the chicken and in the owl. So this is kind of an abstraction of the anatomy and the connectivity that we see in the chicken. And you can see that it looks exactly like the Jeffers model. I have from the ipsilateral ear, all the delay lines take the same time, and then from the contralateral ear, we see there is a difference in the time that it will take for the signal to reach the coincidence detector. And this is how the anatomy looks like. And it's the same for the owl. For the owl, we see a lot of different paths. For the chicken, just one. But we can really, if you look at the connectivity properties, you really see the Jeffers model. So this is also something that scientists find very convincing. I asked one of the scientists that presented this to me, why, like, how do you know that the Jeffers model is how these animals compute sound organization? And he said, it's right there. You can't miss it. So this is a slightly different case because what we have is connectivity directly. But I think it's convincing not just because of the connectivity, it's because we see organized connectivity properties that relate to relevant environmental properties. Right? It's not just the connectivity somehow fits, but we see the organization of the connectivity that makes it look purposeful. And the structure that we see works in two ways. It constrains the possible interpretations of neuronal activity, but it also makes it seem very unlikely that this is incidental. We see the nice organization also of the angle is represented according to the location of the coincidence detectors and the nice structure of the delay lines. And I think this is interesting. For this example, the neural activity in the coincidence detector, neural activity in these neurons, the coincidence detectors, is actually modulated not just by the interneural time difference, but also by the magnitudes, the amplitude of the sound. So if we would just look at neuronal activity, we would miss completely this beautiful structure. And we would say maybe this is a neuron that has context-dependent computation, or maybe it's representing multiple things at once, etc. But by looking at the connectivity and anatomy, we get a representation that's clear and hard to argue with. Okay. So if you accept that anatomy is relevant, what are the implications? I think for philosophers, this is what I take home from this. I am happy to talk about it during the Q&A. I think etiology is relevant to the notion of cognitive function used by neuroscientists. We need to look at the history to identify functions. You can make arguments that it doesn't matter in principle, but in practice, if you want to identify the functions, it has to matter. And for scientists, I would say anatomy may not be useful for every brain area, but it's just one indicator. We have development, genetics, variability between subjects and species. There's a lot more to brains than just neuronal activity. And anatomy is helpful in individuating useful units, but of course, it's not the whole story. We need a convergence of evidence. If I haven't said that until now, obviously, it's not just the anatomy. Although I think it didn't receive enough attention so far. That's it. Thank you very much.
We take a 2-minute break, so in case some of you must leave, and then we'll reconvene for questions. We'll have a half an hour question.